This method can be used as a diagnostic tool for cancer to analyze the circulating tumor DNA. This technique interrogates multiple genetic alterations in regions of cluster mutations in a single reaction. This helps to save precious patient samples. This method can provide insight to tumor burden and its mutational profile. It can also be applied to other molecular analysis as an alternative to qPCR to obtain an absolute quantification of nucleic acid target sequences.
Collect blood samples in 7 milliliter EDTA tubes. 2 tubes are recommended to collect around 4 milliliters of plasma. Centrifuge the blood samples for 10 minutes at 820 times gravity within 3 hours of the blood draw to separate red blood cells, white blood cells, and plasma.
The time between the sampling and the centrifugation is critical to obtain high-quality cell-free DNA not contaminated with DNA released from white blood cells.
Following centrifugation, use a serological pipette to collect the supernatant without touching the white blood cell layer. Around 2 milliliters of plasma are usually recovered per EDTA tube. After transferring the plasma to 2 milliliter tubes, centrifuge the aliquots at 16,000 times gravity for 10 minutes at 15 degrees Celsius to remove cell debris. Carefully collect the plasma without disturbing the pellet and transfer to 2 milliliter cryogenic tubes.